Hey everyone, my name is Mohammad, and I'm excited to talk about a bioinformatics project that I did this semester in investigating a novel toti-like virus. Okay. And so Serratus was able to detect this novel virus based on the detection of the PAM-ID sequence, or the core catalytic uh, sequence of the RNA-dependent RNA polymerase in RNA viruses. And so Serratus was able to detect this new virus, which is given this taxonomical unit identifier. And this specific novel virus was the sequences that may encode for this virus were detected in the SRA runs of 21 SRA runs that originated from either human or mouse origin. So the sample came from either human or mouse. And so we aim to, study and investigate the biology and the genome of this virus. And so to assemble the genome of this new virus, we went back to the Serratus and see the PAM-ID sequence, or the core catalytic region of the virus, and using tblastn to search the Logan contig the Logan assembly files of these SRA runs to see which contigs have uh, the sequence that encode the RNA sequence that encode uh, the virus. And we found these 7 contigs, which are shown here, all aligned, and they're all pretty short. It only covers like 387 base pairs. And unfortunately, we're not able to extend the genome. And so we focused on the longest of these contigs, which covers all the others, which is the one here, and using ORF-finder to predict a possible ORF in the contig. And then we ran the amino acids, from the putative ORFs in BLAST-P, NCBI's protein BLAST. And we found these hits, which are corresponding to toti-like viruses. And so that's how we determined that this virus is possibly a toti-like virus. And so because our genome is really short, it's only a partial sequence of the RdRp protein based on the BLAST-P hit. So to represent this genome, we chose to align it with the top hit, the genome of the top hit from BLAST-P, which is this diatom colony-associated dsRNA virus 3, which is a toti-like virus. And using a local pairwise alignment tool, we're able to map our new genome into the ORF2 of the toti-like virus, which encodes the RdRp protein. And when we put it in an AlphaFold and did a structural prediction on this partial RdRp sequence, we're able to see the conserved motifs in the palm region of the RdRp, which is motif B shown in green and motif C shown in red. And that further proves that this region is indeed a encoding for RdRp. And so what is totivirus? So Totiviridae is a family of viruses that are known to infect fungi and protozoa. And it's a double-stranded RNA virus. And interestingly, many of them are known to also infect several human parasites, protozoan parasites. For example, this picture on the left here is showing a disease called leishmaniasis, which is a skin disease caused by this protozoan uh, protozoan parasite from the group of Leishmania. And so what's pretty interesting is that the toti a genus under Totiviridae called Leishmania virus. The members of Leishmania viruses can infect Leishmania parasites, and this infection can actually cause an increase in pathogenicity of Leishmania infection in human, which is very interesting. And Leishmaniasis is the parasite Leishmania. Parasites are transmitted by this vector sandfly, which that will bite on humans to suck up blood for meal. Uh, and so, and this is relevant with our observation in that the 21 SRA runs that have uh, uh, the viral genome, most of them came from plasma or blood samples. So that indicates that, so we can, make an assumption or make a prediction that this new virus, this new toti-like virus, might be related to Leishmania viruses because the transmission of Leishmania parasites are through there's a contact with blood in its transmission. However, this is in contrast with the top BLAST-P hit, which shows that the partial RdRp sequence is very similar not to Leishmania viruses, but instead to viruses similar to toti-like viruses that are derived from environmental microbiomes. And so due to this unique contrasting observations, we're provisionally naming this virus as Diatototivirus from diatoms sanguinis, which means of blood. And so to answer the question whether this virus is more related to environmentally derived toti-like viruses or Leishmania viruses, which are related to blood, we're doing a phylogenetic analysis on the partial RdRp sequence of our new virus, Diatototivirus, and doing muscle alignment with with several other viruses. And for outgroup, we're choosing Serratus-defined, which is a member of a Chrysoviridae family. And then we also picked several representative viruses from other genera in the Totiviridae family, including Trichomonas, Leishmania, and several environmentally derived toti-like viruses. And so as we can see here, our virus, Diatototivirus, is more related to the environmentally derived viruses, which is very interesting. And also we can see from the general structure of this tree, it is more or less consistent with a previous analysis, phylogenetic analysis on the Gabriviralis order. So it's the level above Totiviridae. As you can see, there's Leishmania viruses being a sister taxa of the environmentally derived viruses. And as you can see here, Leishmania viruses is a sister taxa. I mean, not directly a sister taxa, but it's closely related to the diatom colony associated viruses. Also, we also see that Trichomonas here as a sister taxa to the whole thing. And then we also see Trichomonas viruses here as a sister taxa to both Leishmania viruses and the diatom colony. associated viruses. And so from our phylogenetic analysis, we can infer that Diatototivirus sanguinis from our sample may have actually come from not from Leishmania-related events, but instead may have come from exposure to environmental microbiome. So, for example, maybe it's from seafood diet because many totiviruses are known to infect fungi and protozoans. They're associated with fish, with marine arthropods and insects, and also this virus may also have come from aquatic activities. However, it should be noted that our analysis is very preliminary, and I would say it's a very low confident uh, analysis because our genome is not complete, where we only had a partial, really short sequence of the RdRp protein, which, as we saw, was kind of we could capture like the general pattern of the phylogenetic of the phylogenetic tree, but I believe that further investigation is needed to get a more complete sequence of the genome, and so we can do more reliable phylogenetic analyses, and we can also explore many more interesting biology of this novel virus. So, thank you.